Hey guys, this is Mr. Herbst here. Today's focus is going to be on nerve impulses in anatomy. I had a few requests to add my face in here, so hopefully it doesn't scare you off too much. We've been focusing on this thing called a neuron, which can send an electrical impulse, otherwise known as an electrical current, through an axon. When combined with other neurons, it is the foundation of how your body transmits quote-unquote messages. Here I have a diagram of what a neuron looks like. This part right here, that long extension, is the axon, and so your neuron actually has the ability to send an impulse down the axon like this, and so when we get a clump of axons all working together, that's what we call a nerve. So a nerve is a bundle of axons. You can think of that kind of like analogous to an electrical wire, where electrical wires can send signals. So all along your body, with this diagram here on the right, you have all these nerves, and all of these work kind of like an electrical wire. They're bundles of axons that send electrical signals up to your brain for them to be processed. So how does a nerve impulse fire? What actually is a nerve impulse? Well, it actually is a diffusion of certain ions across the cell membrane of the neuron, and that typically occurs at these nodes called between the Schwann cells. So here I have a Schwann cell, that's this right here. In between the Schwann cells right here is a little node, and that's where we don't have any myelin sheath there, and so we can get ions exchanging at that point right there. And the ions exchanging is actually what's going to cause that neuron to fire. I have this diagram over here on the left. This is one of my favorite diagrams to look at. But you can actually see here that a cell, this would be a starting cell at rest, that has a negative 70 millivoltage of membrane potential or voltage potential. And that at any given time, a neuron has the ability to make a nerve impulse fire, produce a little bit of electricity, and then it'll go back to rest. So all the way up here, here is 0. You can see that a neuron can actually produce, well, on this graph it says 30, but some neurons can produce about up to 50 millivolts. So for the next few slides, I want you to imagine one thing. It's going to be a little bit difficult to think about, but here I have the whole neuron. This whole thing is a neuron. I'm going to be referring to this little section right here, the node, what actually is occurring at the node. Okay, so imagine that this picture here this is the membrane, you can see the phospholipid bilayer. This whole picture here is occurring right there at that little node. So here I have, on the bottom is the inside of the cell, and out here is on the outside of the cell, and in between them, separating them, is a membrane. You can see all along the way that we have these proteins. We have the sodium-potassium pump, we have some potassium channels, and we have some sodium channels. Now what does it actually mean to be a potassium or sodium channel? Those are proteins that will only allow a certain ion to pass through. So potassium channels, what do you think they allow through? That's right, you guessed it. They only allow potassium to go through. Potassium is these little squares right here, and then sodium will be represented by these little circles right here. So the resting potential occurs when a cell is not firing. That's important to know. So the cell is at rest here, but a few complicated things are actually happening that would make you think the cell is not really at rest. But again, what it means to be at rest if you're a nerve cell is that you are not firing an impulse. The sodium-potassium pump will pump out 3 sodium ions for every 2 potassium ions that it pumps in. So we're going to get this higher concentration of sodium on the outside, because sodium's going out, and we're going to get higher concentration of potassium on the inside, because potassium's going in. And we also have some negatively charged ions on the inside, which we're not going to concentrate on too much, but they do play sort of an important role later on. This will give a cell a negative charge overall. And what do we call that negative charge? We call that being polarized. And so the cell, at any given moment, is negative on the inside. It's polarized, and it has a voltage potential of negative 70 millivolts. So it has the potential to produce volts. So I'm going to go ahead and walk through what's going on in this diagram. You can see here that I have the sodium-potassium pump. That's this protein right here. It is pumping out these sodium ions right here, and it's pumping in these potassium ions in here. So overall, we are getting more potassium coming in and more sodium going out. So you can see you can see that there is more of these circle guys on the outside than on the inside. So overall, because we have 3 sodiums going out, for every 2 potassiums that goes in, we get this overall net of negative on the inside. Also, we can thank a few of those negative polyatomic ions for allowing the cell to be negative on the inside. But overall, a cell is going to be partly negative on the inside and partly positive on the outside, at least in relation to each other. So at any given moment, the cell can sort of screw up things, and all this sodium can come rushing in, and all this potassium can come rushing out. And that's exactly what we're going to talk about in a second. The next thing that I want to talk about is this thing called the threshold potential. Certain stimuli will cause some sodium channels to open. So some stimuli will actually begin these sodium channels to open, and that'll allow sodium to rush in. When sodium rushes in, the cell sort of depolarizes, because it's sort of like we're having that positive come in contact with the negative and sort of neutralizing it a little bit. So it's beginning to depolarize a little bit. The bigger the stimulus that we have, the bigger the depolarization. So when a neuron depolarizes to negative 55 millivolts, we get this action potential. That's when we actually get the neuron to fire. So when we depolarize to negative 55, the neuron is going to fire. You can sort of think of that like this. Here I have a thermometer. Let's say that it's a comfortable 80 degrees outside. You know, you're comfortable, but it's nighttime. This temperature starts dropping, and you can sort of feel the temperature dropping, but it's not uncomfortable yet. And you're sitting outside, it's getting darker and darker, and the temperature starts dropping, and eventually it reaches 60 degrees. And that's when you start to feel, man, you know, it actually is kind of cold, so you run inside. So it's sort of like that. The cell depolarizes a little bit, then bam, it fires an action potential. And what we call that is an all or none response. Basically, either a cell will fire an impulse or it doesn't. It's either on or it's off. It's kind of like a light switch. It's either on or off. It has no in between. There's no somewhat firing, or there's no somewhat at rest. It's either you're at rest or you're firing. That's it. 2 choices. So I'm going to go ahead and walk you through here briefly actually what's happening when a cell fires. I thought this diagram would be easiest to look at first. So we're going to, again, the sodium channels are going to open. We're going to get some sodium rushing in. And as that sodium rushes in, it's going to depolarize the cell. And if it depolarizes enough, bam, we are going to get an electrical current being fired. That's called an action potential. The actual exchange of that happening is called an action potential. So sodium rushes in, which is going to cause the cell to depolarize a little bit. And eventually, it's going to depolarize so much that the cell needs to repolarize. How does it do that? Well, it has potassium rush out. So sodium rushes in, potassium rushes out. So go back to this diagram here. So the cell is going to depolarize by sodium rushing in, and then eventually we're going to get to this point where we want to repolarize the cell, so we're going to have potassium rush out, which is going to help to repolarize. So this diagram here, I took all the little ions off of here, but you can see that the sodium rushes in, and then eventually we're going to have this impulse being fired down that way, down this direction here. And then we're going to have some potassium rush out when the cell reaches a critical point, and that's going to help to repolarize the cell back to normal. So eventually the sodium-potassium pump kicks in to bring the cell back to its normal resting state. So here I have this diagram again. We're starting here at negative 70 millivolts. So we begin to depolarize, and then bam, when we reach that negative 55 millivolts right here, that critical point, boom, we're going to get that action potential to fire. We're going to get those sodium gates to just rush, just dump tons of sodium in. And the cell is going to produce a voltage right here at the top. This is a positive 30 millivolts. And then those sodium gates are going to close, and those potassium gates are going to open, which is going to help to repolarize the cell back down to, well, you can see there actually is a little bit of dip. That is actually a point where the cell begins to what's called hyperpolarize. What does it mean to hyperpolarize? Well, it polarizes too much, and that happens because the potassium channels right here tend to take a little bit longer to close. So they stay a little open a little bit too long, and we get a little bit of too much potassium rushing out, so that cell hyperpolarizes a little bit. But eventually the sodium-potassium pump is going to kick in and fix all that stuff and bring the cell back to rest. And that's where we can have, at this point, we can have another action potential fire. and then we bring the cell back to rest, and then another one. But all of this takes fractions of a millisecond. I mean, this takes almost no time at all. This happens really quickly. So again, just to review here, when we have an action potential, we're going to have the sodium rushing in. So we're going to get all this sodium rushing in, create a voltage, which is going to force an impulse to go this way. And then the potassium is going to rush out and help to repolarize the cell. And then eventually the sodium-potassium pump is going to kick in and fix all those things. And we're going to go back to rest. Now, one important thing to know is that an impulse jumps from node to node. I had mentioned that this region right here, between the two Schwann cells, is called the node of Ranvier. So right in there. And so actually, you can see that there's this myelin sheath over most of the axon, and we can't have ion exchange at those myelin sheaths because, well, they basically are like insulators. They don't allow for certain ion exchange. So the only place that we can have ion exchanging is right here. So what's going to happen is an impulse is going to jump. It's going to have some ion exchanges. It's going to jump over here, ion exchange, jump over here, ion exchange, jump over here, ion exchange, and then eventually to the axon terminal. Now, one important thing to know again is all that stuff happens super fast. Well, anyway, hopefully that helps to clear up the complicated subject of what's going on as a nerve impulse fires. This is Mr. Herbst here. I'm signing off, folks. You all have a nice day.